Right now, Unit 7, what we should know about Unit 7. Here, very important thing is we have to go a little bit beyond of the content from resource book to understand things. Certain levels, when it comes to the cell, you need to, this is a Golgi. Golgi looks like this, and this is the function of Golgi. That's it. Anyone can understand it. But when you are coming to a very advanced topic like Unit 7, if says, okay, this is the structure and this is how it works, you lost. Because many things are missing in between to understand it. When it comes to the molecular biology, some of the topics, they just give the overview, most of the links between missing. So then one point to the another point, you don't see any relationship. So we need a little bit of, in certain sections, need to understand things. But for remembering what is in the resource book is enough, because it gives the summary of all important things in short. Even if there's enzymes, the enzymes are listed and function are listed. These things are important to remember. Okay, so when it comes to the first part of this lesson, we're going to learn again the structure of the DNA molecule and chromosome and DNA replication. That means how DNA molecule become one DNA molecule become two DNA molecule. So it is very important. If there is a one glucose molecule and two glucose molecule produce, it's a simple task. But the biggest molecule, we can say the probably biggest molecule in the cell, and not only the size, it has a certain order of the monomers. This monomer order cannot change. So we have to copy without changing this order. Just like if we have a book, we have to make a same book without making any errors in the words. We have to copy exactly same book without making errors in the words or without changing the pages. So we can't make errors. So that's the DNA replication is very a complex process. So we're going to learn in the first part, DNA molecule and replication. Now what you see here, electron microscope picture of the DNA molecule. Right. The most important thing, we should have a very clear idea about the DNA molecule. How exactly DNA molecule is formed and what is the structure of the DNA molecule. So in short, what I can show you, if there is a deoxyribose with 5 carbons, sugar, so it has carbon at these points, and carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So carbon number 1 is important for us. Why it is important? Carbon number 1, there is a bond between carbon number 1 and the nitrogenous base. So carbon number 1 and nitrogenous base, there is a bond. I just represent one nitrogenous base like this. This is a nitrogenous base. Carbon number 2 is also important. Why carbon number 2 is important? Because this molecule becomes deoxyribose. Deoxyribose is this carbon number 2 lost one oxygen it has only a H. So deoxy, oxygen lost at this position. We call deoxy. And carbon number 3 is also very important. Why carbon number 3 is important? Because carbon number 3 is responsible to link with another nucleotide. So it can make a bond with another nucleotide. So carbon number 5 is also important. Why it is important? Carbon number 5 linked with a phosphate group. So this represents the nucleotide. And we know there are 4 nucleotides like this. As nitrogenous base change to adenine, cytosine, thymine, and guanine. As a result, we have a adenine, thymine, cytosine, and guanine. So adenine and guanine, these two, they have a 2 rings, and we call them purines. And cytosine and thymine, they have a 1 ring. So we call them pyrimidine. So cytosine and thymine are pyrimidine, adenine and guanine are purines. Now this structure joined with many nucleotide to form strand. So we use this word strand. We call the word strand. How it work? To understand that, we again need this number, carbon numbers. So this carbon 1 is called 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So we can also label carbon in glucose, carbon number 1, 2, 3, 4, 5. But when it comes to the DNA and RNA, we have a special addition for this one, prime. C1 prime, carbon number 2 prime, 3 prime, 4 prime, 5 prime. So how it work now? It work like this. The next nucleotide, the phosphate group is attached to the 5 prime carbon. So this end is a 5 prime then. And here there is a OH group. And there is a reaction between this phosphate group and OH group to make a bond. So now this bond is created to attach one nucleotide with another nucleotide. Hence, we call this phosphodiester bond. So phosphodiester bond link one nucleotide with another nucleotide. So we can add always nucleotide like this way to the end that the 3 prime place, like here again. Again, we can add another nucleotide to the 3 prime place, and we can make a long chain with this nucleotide. So this long single chain is called the strand, DNA strand. And once a single strand is formed, there is another strand formed in front of that, and then they join together. But there is a speciality. Speciality is when one nucleotide strand is formed, phosphate, nitrogenous base. Second one, I can only use maybe 3 maximum just to show the strand. Something like this. This only you can see 3 nucleotide form the strand. Now this end remain as a 3 prime and this end remain as a 5 prime. Now the second strand changing its direction, it's going opposite direction. That means the sugar molecule is like this, nitrogenous base is like this, and this is a phosphate group. Now where is the 3 prime? 3 prime is here. This is a 5 prime. Now this is the strand for that one. Phosphate, phosphate, nitrogenous base, nitrogenous base. Now here 5 prime. Now you can notice the 5 prime of this strand is this side and 3 prime is here. This is opposite now. 3 prime is here, 5 prime is here. So we can call they are anti-parallel. The strands are anti-parallel. So here is the link. So this is a strand 1. This is a strand 2. Now why this kind of orientation is important? Because when it run opposite direction, this provide the best configuration, spatial configuration to form hydrogen bond between this nitrogenous base. So nitrogenous base has OH and H groups. So this OH and H group can make hydrogen bond. Now these two strands going to join from this level with the hydrogen bonds.
So can you explain that anti-parallel strength exactly?
Yes, antiparallel structure is this strand is running 3 prime to 5 prime direction. Okay. Now, if we turn it upside down, this is running 3 prime this way and 5 prime is this way. So you look at the arrows. This is going upward, this is going downwards. So they orient like this position. One is going upward, one is going downwards. The reason is this is the best orientation to make the hydrogen bonds. If we change the direction, it can't make hydrogen bonds. So one strand need to turn the opposite direction if it binds to the next strand, because the hydrogen bonds can make if it is in anti-parallel direction. Otherwise, hydrogen bond can't make. So then these two strands join together by hydrogen bonds. So then DNA molecule, one strand is running 3 prime to 5 prime, other strand is running to 5 prime to 3 prime. That means if I draw it again, this is a one strand and this is another strand. These strands are joined together by hydrogen bonds between the bases. And one is running from 3 prime to a 5 prime, other one is running 5 prime to 3 prime.
Yes.
So this gives some kind of a basic information about the DNA molecule. And as a result, The phosphodiester bond is a very strong covalent bond. So this bond is very strong one, and this bond is a very strong one. In the middle, there's a weak bond of hydrogen. So this kind of two strong bond in the either side and the middle weak bonds resulted twisting of this molecule. So it going to twist. If it is twist, it make this double helix like this. So it naturally going to orient double helix structure as a result of this twisting, and we can still find in the middle the hydrogen bonds between the bases. And we should also remember adenine make 2 bonds with thymine, cytosine make 3 bonds with guanine. And this kind of base pairing is referred as complementary base pairing because it follows the rules always A with T, C with G. A with T, C with G is the complementary base pairing we can find in the DNA molecule. Right, we need this basic recap of the DNA molecule to start the advanced lessons coming up in the next section. So this is something we recap from the past, how it looks like. Okay, now we're coming where we can find DNA in the cells. We can find DNA in different places in the cells. We can find in the mitochondria. We can find in the chloroplast. We can find in the nucleus. That means main station is the nucleus. So we consider the main place of the DNA location is the nucleus. Right. When we look nucleus under electron microscope, you can see this kind of a picture. Why do you see two colors? How do you explain these two colors then? Let's say dark and light areas. Some area looks very dark. Some area looks very light. But I still say the nucleus is packed with the DNA molecule. That means the complete structure, what we see is DNA molecules. So DNA molecule packed inside the nucleus, and we can see light areas and dark areas. Okay. Precisely, we can write instead of DNA, what we see here, dark area are chromatin. Light area, they are also chromatin. Now, what is chromatin? Chromatin is DNA plus protein. Oh, it's not writing well. DNA plus protein. DNA plus protein. And sometimes people try to write DNA plus the name of the proteins. There are different proteins, but abundant protein, common protein name is given DNA plus the common protein in the chromatin. The common protein is known as histone. So people write DNA plus histone because histone is the biggest protein inside this chromatin. But generally, there are other proteins, minor proteins as well. So DNA plus protein is the chromatin. But why still dark and light areas? Why we see dark areas and light areas? Do you know the answer for this?
Staining.
Yeah, this is stained. I mean, electron microscope we use heavy metals to stain. The light microscope, we use like colors, methylene blue or hematoxinil like colors. But electron microscope, we have to use heavy metals like lanthanum. They are heavy metals. They can bind to the biological molecules. And electron microscope, they use beam of electrons. So then these heavy metals represent the a part of the shape of the molecules, and we can make the images. So darkly stained and lightly stained, yes, we can say darkly stained and lightly stained. This is a light stain. Now, why some chromatin stain dark? Why some chromatin stain light? Do you know answer for this one?
Proteins are different.
Mostly proteins of the DNA molecules are same everywhere. Its reason is simply we can say amount of chromatin. That means dark area, the chromatin is higher. The light area, chromatin is less. That means DNA is less. That means concentrated. We can say it's a concentrated. So we don't use this word concentrated. Like it's like a lightly packed. The right word is loosely packed and tightly packed. It's about the packaging. So this dark area has tight packaging. It's packed. It's completely very tightly packed. As a result, we use this term heterochromatin. So these dark chromatins are heterochromatins, and this one is a loosely packed. So they're called euchromatin. The word is euchromatin use. Euchromatin and uh, heterochromatin. That's how we can explain the appearance of nucleus under electron micrograph. And people further trying to explain this, the light area, the DNA is active. That means they are ready to copy, easy to copy because they are a little bit loosely arranged. They can